I bring you greetings from the Dinosaur Soft Tissue Research Institute. We are an institute dedicated to finding as much as possible around the whole globe dinosaur soft tissue and publishing it. We go to scientific meetings, we publish. The work you're going to see today is at a journal right now, and it's going to be published in the month of August. And I wanted to bring you the latest and greatest. It's a little shorter because I know the attention span. What is the old saying? The brain can absorb only as much as the derriere can endure. Yeah? So I'm going to be brief to the point, but I'm loaded with information. What we're going to do is, if you have not received a 3x5 card, and you might want to ask a question, raise your hand and Ruth will get you a card. And if you need a writing utensil too, let us know. Because a lot of the information comes out in the Q&A, and I want to have time to answer all your questions. The Dinosaur Soft Tissue Research Institute is a 501c3 organization. We're headquartered actually here in Texas. And today you have the three founders, Ruth, myself, and Keith, who's our president. Keith Holcomb, stand up and say hello. This is our president. And wonderful, wonderful man, real heart of God. And I love you, brother. And I appreciate your partnership. But he's really helped us to get a solid foundation. And it's an open organization. We're all volunteers. None of us makes a dime. All the money goes straight to the work. And we give everything away because we want the world to hear what has been found. And you're going to be amazed. So you can go on the website, dST.org. I'm going to point powerfully with my PowerPoint thing. But there's the website, dST.org. The paper reprints, so the papers that have been published in journals are available, and they'll continue to be available for free. There are selected videos, updates where we're speaking, and we have great plans to go speak at college campuses all over the country. So we'll give you updates. All our books are free. I have copies of Old Stretchy #1. This was published back in 2013 or 14, I think. And so each family gets a copy. And if I'm not looking, take 2. All right? But share the second one. We want this information to get into the hands of people who need it. Okay? If you want more information about me and the work, that we have a YouTube channel, just go to YouTube and search Mark H. Armitage. And there are videos that go all the way back to the beginning when I first discovered the soft, stretchy tissue, which you're going to see tonight. I was fired for this work. I was running $1 million laboratory at a state university in California, and I lost my position. So if you want to hear about that, just Google Triceratops lawsuit. And you don't even have to add my name. It all comes up. And so we really thank you. Now this talk, The Soft Side of a Mean Dinosaur, is about a kind of a new dinosaur called Nanotyrannus, which maybe you've heard of. It's a controversial animal. And the reason we went after it is because it already has a controversy. So if we can show dinosaur soft tissue in a dinosaur that everybody's already aware of in paleontology, we figure we've got a winner there. But this whole presentation on Nanotyrannus is on dST.org, and it's lengthy. There are 3 actual 30-minute videos, I think, or 45-minute videos. And I'm going to let you do that for homework, okay? Because it's all free, and you can go on to dST and see that. Today I want to show you the latest work, which I think is going to really excite you. Then when I finish, I'm going to be abbreviated, but it's going to be powerful. We'll take a short break. You can go to the restroom, come up and handle the bones. We've got a whole slew of bones here, all of which have yielded soft tissue, and of course our free book. So does that sound okay? All right, let's get going here. I want to talk a little bit about soft tissue, because to understand microscopy as a discipline, you need to understand soft tissue. You just can't take any old soft tissue and throw it under the microscope because it's amorphous. It doesn't really have a shape. You have to tease the shape out of it and work with it. And I'm going to give you some examples of some of the soft tissue that I've worked with in the past. Maybe you've heard of the grunion fish from California. This lays its eggs in the sand, right on the beach where the wave action is, where animals and people and cars are moving. So they're in a very dangerous environment. So these little eggs need to survive. Well, I thin sectioned, and so far I'm the only one to do so, I thin sectioned this little egg wall, and it's full of structure. I'm not going to take the time to describe it all, but you can see different layers of structure in the egg wall that keeps them protected while they're in the sand. But this tissue has to be processed for about 80 hours to get it to this point. That's when you can really image it under the microscope, particularly the electron microscope. This is about 10,000 power. And you're beginning to see that in the outer layer here, besides a sandpaper layer, and by the way, the whole egg is coated in fat. That's a lipid layer around the outside of the egg. But inside that lipid layer, there are crystals embedded in this tissue that give it rigidity. So the more we magnify, the more design and structure we see in these things. But again, that's about 80 to 100 hours just to get that one picture. I did some work with a termite when I was teaching at Azusa Pacific University. And so this is termite intestine. And just like us, in our intestines, we have this brush border. Now, it doesn't look like much here. It just looks like a gray border. But when you magnify it, you'll see that it's full of little fingers that absorb, and you have this in your intestine. So these little fingers do all the absorption. But I want you to notice something. Termites eat wood, right? But they cannot digest wood. Did you know that? A termite cannot digest wood, yet it eats it prodigiously. But it has these little partners. It's a protozoan partner, and they actually embed themselves right in these structures and are pressed right up to the intestinal tissue. So they can pass those nutrients right into the termite. So it's a beautiful symbiotic relationship. But again, 80 to 100 hours just to get that picture. As Dr. Baugh mentioned, and thank you again, Dr. Baugh, for inviting me. This is just awesome. I did my work in parasitology, and I got several journal covers, which I didn't ask for. I just presented the paper, and they published it. But these are parasites, again, soft tissue, which take many hours. So I had a lot of experience in soft tissue before I read the journals that were talking about dinosaur soft tissue. And I was shocked. As a biologist, as a microscopist who works with tissue, I was shocked. But then as I read the materials and methods that they used to get their work done, I realized I'd already been trained in all that. So I had all that training. So it was a natural for me to go on a dig. And I was so excited. We went on a dig. We dug for 3 days in Montana, didn't find anything. And we were looking for long bones. I'm going to show you long bones in a second. Because most of the scientists who find soft tissue, before I started, were finding them in long bones. Why? A long bone, a femur, particularly a dinosaur femur, and I think there's some long bones out there you can see, they're built like brass. Because there's been a big push to uncover the mechanism for preservation, right? How many of you guys have heard of the iron preservation theory? All right, this is a big topic. A couple of you have. And they're pursuing this because they cannot explain the presence of these tissues. These tissues are impossible. As a microscopist, as a biologist, it is shocking to see the level of preservation. And we're finding the deeper we go into the rock layers, back in time, as they say, the better the preservation. Let's talk science. So I mentioned long bones, and most of the scientists that started looking for dinosaur soft tissue were looking in femurs and humerus bones and such because they're encapsulated. The dinosaur bones have a very thick layer of compact bone around the outside of the bone. It's like a little sarcophagus. It's a time capsule, really, with the elements inside. But we were not able to find a long bone. So on the 4th day of the dig, when we had to drive back to Los Angeles, we finally found this horn, 48 inches long, and it took us 8 hours to get it out of the ground. But bone, and I'm not going to spend a lot of time on this, you can see this online, bone makes these concentric circles. It's actually the cells, the little osteocytes that make this bone. The first thing they do is they make a carpet of collagen. You know what collagen is, right? Those of us that are aging, we need more collagen, don't we? It decays over time, right? But it's a very stable molecule. And so they make this carpet, and they all lay down on the carpet like soldiers at parade, and then they cover themselves in bone mineral. They actually bury themselves deep inside the bone. But before they do that, they make little tunnels to all the other cells in all directions, and they send a little, it's called a thread foot, a filopodia. These little structures, they reach out, they go through the bone, through tunnels, and they touch every other cell in the bone. Every single cell in your bone is talking to every other bone, every other cell in your bone. Why? Because they monitor you as you run or carry, right? They're monitoring your bone. These little things are measuring the stress on your bones and accounting for fractures, and they're repairing them. In fact, your entire skeleton is replaced every 12 to 15 years. Did you know that? Your entire skeleton is rebuilt by these little cells. Okay? So the osteocytes, those are important, and we found tons of those. And you're going to see some tonight. But the other structures that I think are more important are the ones that are traveling through the bone. The vessels that deliver the blood, the veins which carry it back to the heart, and nerves. You see this yellow line in here? These are the arteries and veins and nerves that are surrounded by these cells in the bone. All right, everybody got that? It's not that complex, but I don't want to spend too much time on it. So, let me show you what we're finding. These are veins. Now, what we do with a bone is we put it in a weak acid. Remember when you were a kid, maybe you took a chicken bone and you put it in a jar of vinegar and left it for a week and you brought it out, it was all floppy. That's decalcification. The vinegar is a weak acid. So we use a weak acid, we soak the bone in it, and then all the tissue structure that's in the bone falls out on the Petri dish. And that's what you're seeing here. These are all veins that were up in the 3D bone, right? And they dissolved out, and now they're laying on the bottom of the Petri dish. So we find veins, and we find vessels. But we also find vein valves. What's a vein valve? Well, when your heart pumps, it's pushing blood through the arteries, right? The blood vessels. And then when the blood returns to the heart, it comes back through the venous system, the veins. Well, there's little valves in there. Why? Because the blood pressure is lower in the veins. So when it pushes blood up, the valves open, the blood gets pushed up, and then they close. They're holding that blood in place. And so it ratchets it up. Each time they open and close, open and close. We have found the actual vein valves from the veins. Here, I don't know if you can see it, they're stained kind of pink. I stained these vein valves with an RNA locating stain. That pink color, that's all RNA. You've heard of DNA, RNA, deoxyribonucleic acid. It's a single strand. This is all single strand DNA that's returning a signal. You're going to see more of that later. But if you look at this picture, you see the ring of the valve, and look at these little vellums of tissue. There's actually, it's a bicuspid like your heart. One goes up, one goes down. So that's how they open and close. Now I want to show you a video. If you look really quickly, you'll see those little things swimming around in there. Play it once more if you can. Yeah. See those little dots in there? Those are bacteria that were in the bone, soil bacteria, that came back to life because of the water. They make spores. Bacteria go into spore stage. And so they're swimming around inside of this vein valve, and they got stained with the RNA stain also. So they're glowing red, just like the vein valve is. See them glowing red? So that means they couldn't get out of the vein valve. It's actually closed upon them. So it's a fully functional, intact valve. Do you understand my meaning here? This is a fully functional valve that's supposedly 68 million years old. Okay, so this little piece of tissue is 1/100th the diameter of a human hair. How can that still be here? That should have decayed to nothingness. Apoptosis, programmed cell death, should have rendered that to dust in a matter of years or even decades, right, at the outside. So there's no way, this is impossible, that we're seeing these things. In fact, I was actually able to use a regular tissue stain, kind of like food coloring. And this tissue acted like a normal tissue, and it took up the stain. Look at how dark this is. That's a venule valve with a stained cuspid in it. So it behaves like regular tissue. Shouldn't be, but it is. What else are we finding? We're finding nerves, ladies and gentlemen. This is a nerve from a condyle, which is a softball-shaped bone at the base of the Triceratops skull. It's called a condyle. And it was full of nerves. And I've found nerves now in just about every bone we've looked at. Now, nerves are collagen with axons. There's neural axons in there. And they're wrapped in myelin. They're wrapped in fat. And you can actually see the wrapping. See the wrappings? And I'm going to show you this in more detail because I thin sectioned them. This is what we do in biology. In order to understand the structure, we thin section things to see the structure. Here's a little higher magnification. And the neurosurgeon who did my neurosurgery, I sent him the paper to review it first, and he was astounded. This is a guy who knows nerves. He operates on them all day long, every day. These little white bands in here, these are called the bands of Fontana. You can see them faintly all the way across. Even down here, you can see some of them. That's a dead ringer for nerve. If you have bands of Fontana, you've proven that you have nerve. So we're finding these structures. And what I did is I took a chicken bone and I dissolved it in acid and I got a nerve out of it. That's a chicken nerve. Compare it to the condyle nerve and to the vertebra nerve. They look almost identical, don't they? The coloration's a little different. That's an artifact of the polarized light microscopy. But structurally, these things are identical. But it gets better. When I thin sectioned the chicken nerve, I took off the top layer of the nerve. You can see the myelin sheath. See that fatty covering? So that's what wraps the outside. But inside of that is a crosshatch, almost, what would you call this, a tartan weave or something? I'm not a textile person. But it's a weave. See the cross? It's going this way and it's going this way. That's in the chicken nerve. Look at what I found in a decalcified Triceratops horn slide. This was horn. This is the same wrapping. It's just come undone. Do you see that? And here it is in polarized light, so you can see the same kind of colors. See that? So we are circling around, I think, one of the most important finds in paleontology. What I also did is I thin sectioned the nerve further. Please note that the nerve that I thin sectioned all the way through that you're about to see was a Triceratops nerve from the vertebra. So here's the chicken nerve when I first started sectioning it, and then I cut all the way through it. And you can see all this structure in here. But the other thing you can see is these band markings that pretty much go all the way through. So I know I have nerves. There's no question about it. That's one of the things we're finding. The final thing that I want to show you today, I think is most compelling, is we take these bones and we make thin sections of them. We make nice thin sections, about half the diameter of a human hair, and then we can examine them under the microscope. And this is a thin section of bone. But notice all the dark areas in there. You see all those dark areas? Most paleontologists don't see that because they flush the bone out before they thin section it. And as they're processing, right, they wash all this stuff out of there. We did not wash our bones. And you're going to be amazed at the results. All of these are vessel canals. By the way, I'm going to show you guys something, and don't say a word of it, okay? This is just for you. We're finding nematodes inside the bone, in every channel. Why? There's food in them there hills. There's still food in these bones, and they're chock full of these organisms. But these are blood clots. This is clotted blood inside the bones. And we have found this in 4 different individuals now. We've examined the bones through thin sectioning of 4 different dinosaur individuals. They're all clotted. When you put this under a high-powered microscope, you can look at it in polarized light, and you'll see some coloration that comes about because of the collagen. The collagen will return a red signal or a green signal. Here you're seeing green. But this red here, those are the blood products inside the canal. Then I put it under a fluorescent microscope. It's very complicated. But I hit it with blue light. I'm using only blue light. Look at how clear the blood products are in fluorescence. And the bone changes, doesn't it? The bone is glowing. Why is the bone glowing? Because all the proteins in there are being hit by the blue light, and they're giving off light. But the other thing I want you to notice is how the blood has stayed in the canal. It has not crossed into the bone. Why is this important? Because the scientists who are working on dinosaur soft tissue now claim that it's the iron that preserves the tissue. It's the iron. But the iron never left the canals. So how is all this tissue preserved if the iron didn't get into them? See how foundational this work is? I'll show you some more examples here. And this one's even more dramatic, because these are red blood cells that have crystallized, and you can see the individual ones. They've crystallized. So polarized light, blue fluorescence, no spread into the bone. I have other pictures that show this even better. What I did here is I worked with a Triceratops vertebrate bone, a vertebra bone, and I thin sectioned it. Here's the clot. Notice that the canal is almost completely occluded. There's a little open space. Most of them, there's no open spaces in the canals. It's just solid blood. Then I put it under UV light in a microscope and look at the dramatic pictures that show that this iron stayed in the canal. It didn't go out into the bone and preserve any of the tissues. And so this is what's being published right now. This is from Horn. We also found it in rib and frill, but 4 different individuals, and they all have clots. Now, why is this significant? Well, scientists, doctors have found with drowning victims that when a person or an animal dies from asphyxiation in water, they're blood clots, and their bones are full of blood clots. And there's no other dying condition unless you have a clotting disease, a malfunction in your clotting cascade. There's no other trauma that produces those kind of blood clots in your system except for COVID. All of a sudden now, here comes COVID, and what is it doing to people? It's clotting their blood. But this is hard scientific proof now that these guys died by asphyxiation in water. Now, these things were in a cement mixer of mud and water. They were torn limb to limb, but they drowned in the process. This is one last picture about a blood clot that I want to show you, which is taken from a 145 million year old Camosaurus. And what you can see here are 2 clots. Here's a canal. You can see the rings around the canal. Here's another canal. It's all clotted. And notice that the clot is infiltrating into this Volkmann canal, which is a connection. These are like steps in a ladder that run through the bone, okay? And they communicate by passing blood through. And look at all these individual blood cells. I'm telling you, the deeper we go into the preservation record, the better the preservation is. This now is a vertebra from a Devonian fish, supposed to be 400 million years old. Look at all the red blood cells in here, individual red blood cells. So folks, we need your help. We got to continue finding this stuff. We've got to continue getting it out there for people because they need it. People need to hear this. So I'm going to end here. I know that's short, but I want to give you a chance to come up, get a book, look at the bones. We'll take 5 minutes, and then we'll do Q&A. Anybody got questions for me?
And we want the questions to be on YouTube as well. So Dr. Armitage, would you please come back to your podium?
Yes, sir.
And who has the first question? All right.
By the way, can you see the eagle over my shoulder on the tree? That's Seattle.
Doug, you have a question?
Yeah, I was wondering what kind of dinosaurs produce the most soft tissue? Is it big ones or little ones or different types, or is it location, area where they're at, or Where do you find it mostly?
Well, there are typical soils that do produce more of the sandy soils, do produce better fossils, better soft tissue because they believe that the moisture has been wicked away by the sandy soil. So certainly sandy soils. Right here, though, in Texas, you've got the Permian red beds. And the Permian is very old. And I'm not going to say a lot about it, but I can tell you it's better than what you're seeing here today. So we can't wait to get that going. But no, in answer to your question, it's ubiquitous. I predicted back in 2013, and a lot of people laughed at me, that soft tissue in the fossil record was the norm and not the exception. And that's what they're finding. So large, small, across taxa, so many different taxa are producing these. So yeah, I think the sky's the limit. I can't wait to try to get some Stegosaurus bones. That one's never been done before. And so there's tons of work to do, and we need helpers. So if you want to volunteer with dST, come on in.
This is incredibly, incredibly exciting. We're having our cake and eating it too. Next hand for question. Yes, two right here. Yes, ma'am.
How old did you say that dinosaur was that you found? How old was it?
Well, they say that they're 67.5 million years old, the Triceratops. But then they get older than that because they go back in time, supposedly. 67.5 million years out to 400 million years or 300 million years.
Yes, my question is more on a teaching aspect. So I'm a kindergarten teacher for a Christian school, and next semester will be a dinosaur unit. Do you have any suggestions, because we have to be very, very careful with how we teach this unit, right?
Yes.
You have any suggestions, any books that would be great for me as a teacher to teach on this? Because, I mean, this couldn't have come at a better time, really.
Yes.
There is one book available, but I have it.
Are you in a public school setting?
Christian.
Oh, well, fantastic. Get the books. How many students do you have?
I have 11 right now.
So we'll give you 11 books for your students.
Oh, thank you so much.
Okay? And thank you. But use the website. In fact, I want to show you one more video. So Mark, where are you? Can you come on up here, buddy? When I found the horn, I cracked it open. It was so eroded. It was full of plant roots. It was full of, in fact, we did what's called a blast DNA analysis where we tested as much DNA as we could find. We found the DNA of 30 organisms in the bone. But when I cracked it open, so it was heavily weathered. When I cracked it open, there was a sheet of tissue adhering to the bone. It was that collagen carpet that I was talking about. And here I am stretching it like a piece of taffy. So this I peeled off the bone, put it in a petri dish, and stretched it right away. So this was the condition that I found it in. If you show them this video, this is on dST.org. Okay. All of our videos are up there, so you can show them the videos. But text me in the middle of class and say, hey, Mark, I got this question. What do I do? I'll answer you.
How many more questions do you want to take?
As many as you want.
All right. All right.
Whoops. All right.
We favor the youth because we all used to be there. Some of us have lost it.
One more.
Yes, your question, sir.
Get this one right.
Okay. Is the Nanotyrannosaurus Rex real?
Is Tyrannosaurus Rex real?
Is Nanotyrannosaurus?
Okay,
the small one.
That's
the small
a great question. Nanotyrannus is controversial because a lot of people think it's a juvenile T-Rex, a young T-Rex. But there are problems because the teeth are different, the jaw is different, the hips are different. Nanotyrannus, they think, could run 60 miles an hour. It was 21 feet long and 12 feet high, with teeth that are double serrated. So just like steak knives, it could slice you right in half. So that's why I call it a mean dinosaur, because it looks mean. I think it's a different dinosaur, sorry. I think it's a different dinosaur and not T-Rex. But there's a whole group of people who say, no, this is T-Rex. So it's controversial right now. But when you come to my lab and you learn how to do this, you'll show the world that it is a new dinosaur. All right. Sorry? Come on up to Seattle. We'll get you trained.
All right. Next question here.
Hi, Dr. Armitage. Brother Jonathan here. I was just wondering about the Devonian specimen that you were talking about just near the end. Did you do that specimen? Did you find those red blood cells yourself?
Actually, my colleague Jim Soliday, who's also a microscopist, he's in Southern California, and we're sending specimens back and forth to each other. And so he found that blood specimen. And he also found that the last clot that I showed you with the orange background, the 2 clots in the Volksman Canal, those were his pictures also. So he's part of our group.
Until last year, we discovered a larger portion, the last meal, of a stegosaurus, and it has over 1,000. Wow, that's more important than what I'm saying. Wow. That it has over 1,000 gastroliths in it, so I'm very eager for you to see it. And so what we're seeing now.
This is me peeling another piece of the soft tissue right off the bone. Now, I thin sectioned this, and I did it on a machine called a cryostat. Cryo meaning frozen, stat meaning fast. When you go into the OR and they remove a malignancy or something, they take it into pathology and they fix it in a fixative, and then they section it on a frozen section machine. And so I thin sectioned this, and it's full of cells. You can see all these cells in the thin section of this tissue, and it made the cover of American Laboratory, which incensed my employers at California State University, Northridge, because it's soft. I didn't have to preserve it. I just froze it and cut it, just like you take something out of the freezer and cut it. It was in this state.
Astounding.
It is astounding.
By the way, when I introduced you, I said your work had been featured on the cover of 6 journals. My associate back there Googled and said, you're being very conservative. It's 11 scientific journals. The cover has featured your work, correct?
Well, I had a question. These biological molecules, they have a half-life to them. Like a half-life of DNA, for example, is 521 years. Is it possible to mathematically model the molecules out and calculate their age through their half-life? And
Well, I'm sure
Now that's an excellent question because he's a biotech, by the
Yes.
way.
Well, you're asking me questions above my pay grade. But yes, there is molecular work being done. In fact, Dr. Mary Schweitzer has used some very sophisticated microscopy techniques to target individual molecules. And it's fun. It's called immunohistochemistry. I don't expect you to memorize that. But you heard the word immuno. So you're thinking immune system. Your body produces antibodies to antigens that come in. And so they're like Lego blocks. The antibody and the antigen hook together like a Lego block. Now on the other end of the antibody, you can put a dye that glows when you hit it with blue light. So you use these antibody-antigen reaction complexes to target individual places in the tissue. For example, the COVID virus. You've heard of the spike protein. You've seen those diagrams, all those spikes sticking out of it. Those are Lego connection points. That's how tissues connect to each other. That's why COVID is so effective at entering your red blood cells, because it fits right over the spike protein on your red blood cell. So we can take advantage of that and target individual proteins. And so Mary Schweitzer, if you look at her work, she's found actin and tubulin, and those are structural components of the cell. We call it the cytoskeleton. You have a skeleton. Cells have a skeleton. In fact, there's proteins. There's a walking protein that can actually pick up something and walk down this actin filament in a cell and carry the box that it has. It's like a UPS box. It even has a tag on it and tells the protein where to deliver it. And it physically walks. It has legs that walks along this actin filament. So she has used very sophisticated microscopy to find these individual molecules. So this is being done.
And is incredibly exciting. Wow, wow. One more question from the back 40. Anyone over here? All right, these are the scholars, I guess. One
Patty-
more
Patty
question,
has a
right,
question.
right here, and we'll terminate with this question. Yes, ma'am.
So the question that I have is going to be very elementary. So when you're going to this Colorado for the dig, and there's a bone already there, or you think there is there.
At our site, from one tiny little bone the size of my little finger, we've excavated components of 18 different dinosaurs already. But it is a private dig on private property.
Okay, so from that, Mark is going to be going there as well to collect soft tissues. I'm going to ask you a very elementary. How are you preserving the tissues from the dig? Are you cutting it, or how are you being able to collect your specimen in order for you to go ahead and study?
Yes.
Good question.
Good question. Preservation. It comes back to that word preservation, doesn't it? It is important when we take bones out of the ground for study in soft tissue to take out a bunch of the matrix or soil with it. And then we bag it, we triple bag it. So we really don't do anything initially. We don't let it sit in a hot car because that would evaporate all the moisture. These things are full of moisture. And we want to keep those conditions as long as possible until we can get it in the lab, out of the matrix, and into the fixatives. The formaldehyde you've heard of, glutaraldehyde. There's other fixatives that we use. And that solidifies the tissue so that it doesn't fall apart any more than it already is. So we use a fixative, and then we go from there. We decalcify the bone and take all the tissues out. But great question. So come to the lab, and I'll teach you all of it.
You can tell there's
I need more workers.
There's a warm spirit of inquiry. Oh, here's a lab tech volunteering right back here. He just raised his hand. Yeah, okay. We're going to do this orderly. I've learned from experience we have to do it orderly or there will just be a rush down here and people can't get around.